Get going. So my name is Dawn Van Dam, and I'm the president of 1 Million Solutions in Health. For those of you who have known us for a while, this is the Technology Evaluation Consortium, and this is our Signature Square program as part of the Technology Evaluation Consortium. We have 4 different programs in the Technology Evaluation Consortium, and this one here is your chance to hear about new technologies or new applications for old technologies and give us some direct feedback on those so that they can be made more applicable for you in the pharmaceutical and biotech industry. So I'm going to give you a brief overview and then we'll turn it over to our presenter for today. But as you're listening, we want you to be ready at the end of this to evaluate this through a very brief survey, which we'll give you a link to as well. This is also being recorded so that if there are other people in your organization who you feel this technology would be appropriate for, we would absolutely love you to share this with them and get them to evaluate the technology as well. So welcome everyone today. We have asked most people to sign in with their initials just so that you can maintain your anonymity, and even when you want to talk later, all we'll hear is your voice. We won't know your name or the company that you come from. So I will give you an overview of the Signature Square program. The technology we'll be looking at today is an ultra-sensitive biosensor. And typically, if we did not have the organization to present the information today themselves, we would give you a presentation ourselves based upon the information that's available, and then we would ask you to also give feedback on this technology. But today we do have the presenting company or organization with us today to do that to the information. So they will do their vendor presentation. There will be an open time for question and answer, which is why we'd love to get everybody on audio. And then we'll have a closed session discussion where there's maybe some things that you don't want to mention in front of this organization or you want us to probe for additional information afterwards. And then we'll send you a link to the survey and get you to answer that nice and quickly. Okay, so moving on to the next slide. 1 Million Solutions in Health. We're all about sharing ideas around the globe to improve healthcare, to accelerate life sciences research, and to bring patients and consumer-focused ideas and solutions forward. We do that through things like this Technology Evaluation Consortium. We also hold webinars, do discussion forums, live events, publish articles, et cetera, et cetera. And we're here to help academia, government, entrepreneurs, and businesses share and accelerate the adoption of science around the world. We have over 5 million people who we touch, so we have a lot of people that we can reach with new science that's available. Why is this important in particular for the pharmaceutical and biotech industry? We did a survey back in 2013, and we asked the industry, we had almost 100 people respond to this survey, what are your key concerns in adopting new technology for the first time for your organization? And you can see that people said things like cost, internal competency to use, availability of the service provider, but the #1 thing was validation of that technology. So this Signature Square is the first step in a validation process where you get to learn more about the technology, and then you get to give direct feedback to this, the sponsor of this this event, the presenters of this information, and then it's an opportunity for you to share with us your feelings and your thoughts and how you see that this would be utilized in your organization. And I'm just going to ask a quick question to Kevin. Kevin, if you can just recheck the number again, because people are saying that when they dial in, they get kicked out. So if you could try it yourself separately, Kevin, and then maybe perhaps put in the correct information, that would be terrific. So, what we do in this evaluation is oftentimes we will send you an information package beforehand. We sent through some information about this technology in order for those of you who are interested to be on the session today for the Signature Square evaluation. We hold a webinar, which is what we're doing right now. We record that, and then it allows you to evaluate all life sciences applications or applicants who submit into the Signature Square. We also have a version of this for healthcare practitioners, et cetera, but this is the version that we're using for you guys in life sciences through the Technology Evaluation Consortium. So it allows you to rank technologies on a number of criteria based upon 2 dimensions. One is the readiness of that technology for utilization in the industry. So we call that the market or partner readiness. And the other is the value proposition to the marketplace. So, and then we issue a summary report to the sponsor or vendor today, outlining the scores and that you have provided by the industry. And all of the information inputted is anonymous, so no comments will be attributed to any particular person or company who has responded. So you can feel free to comment both in the session and also afterwards in the survey. So what, in the end, this produces is a ranking, and the technology gets ranked as either an upstart, a blockbuster, an innovator, or novel. And then we get to kind of see how it ranks versus other technologies that we've been looking at. The benefits for you as the industry is we are going to be bringing you more and more technologies to evaluate, so increase the flow of technologies to be evaluated. We'll sort of do some pre-screening and kind of triaging for you. And because the industry has, you know, reorganized and downsized a bit over the last number of years, this is a way to enhance the productivity of the industry, to bring, you know, more technologies to look at so that you can incorporate what makes the most sense for you as you're looking to bring more efficacious and safe medicines to the patients who need them. The Signature Square program specifically provides you the opportunity to learn more about scientific technologies, to expedite the process, and perhaps also some of these technologies may be chosen by the consortium to receive a full-blown consortium project later on in the process. So we're very excited by these changes that we're implementing and hope that they bring lots of value to everyone involved. Back in April, we did 2 evaluations, just to give you an idea of what some of the results look like. We looked at Stamina, and we looked at Organ on a Chip from the WIS Institute. And these are the scores that the industry participants provided. And so both of these were ranked as innovators. And this is going to be published over the next while so that more people can know about it within the consortium and share the specific details behind it. But this is sort of some of the end results of what the information looks like. So I can move to the next slide. So with no further ado, I'm going to we're going to figure out how we can get everybody onto audio while we're listening to the presentation. If you do get stuck and you want to say anything but you're not on audio, you can always stick your comments into chat box to myself, and then I'll bring them up later as we go on. But I'm going to turn it over now to our presenters for today, and I'll let them introduce themselves. We have Cihan Yilmaz from Biolom and Northeastern University, and Ahmed Basnani, again from Northeastern University. So with no further ado, I will pass the ball, I believe, to Ahmed first, and then let you get going with the presentation. So over to you, Ahmed, and let you give today's introduction.
Yes. Thank you. Can everyone hear me okay?
Yes, we can.
Yes. Thank you. So this is a presentation about a very sensitive, highly sensitive biosensor. This project was started, based on research, directed assembly research, and the NSF, Nanoscale Science and Engineering Center for High Rate Nanomanufacturing at Northeastern, which is funded by the National Science Foundation. And then a group of myself and Professor Vladimir Torshelin, who's also an NIH Cancer Center, Nano Center for Cancer Research Director, got together, and we had funding from the Keck Foundation to fund this specific sensor. And so this was a few years ago, and we had the students that worked on the project became postdocs, and then they spun up a company. And this presentation basically presents the results and summary of the work that's went on since that time. Dr. Cihan Yilmaz, who's a co-founder and also a postdoctoral researcher at Northeastern, will be making the presentation. So I'll give it to you, Cihan.
Yeah. Hello.
Hi, my name is Cihan Yılmaz. I'm the PhD in Mechanical Engineering, co-founder of Biolom. Today I'm going to introduce you Biolom, which produces revolutionary biosensors for highly sensitive, fast, and inexpensive detection of biomarkers in biological fluids. So during my presentation, I will first present you the technology and show the results that we have obtained so far, and I will talk about the potential use of our sensor in pharma and diagnostic industry. So if you look at the challenges in biomarker detection and monitoring, the tests suffer from low sensitivity and specificity, which impedes the accurate detection of biomarkers at very low concentration of biomarkers. So the other challenge is the methods are usually invasive and uncomfortable. For example, you need to draw large amount of volume from the blood, sample volume. So that actually makes it most of the time invasive for animal studies or clinical trials in patients. For example, if you draw a lot of blood from the mice, you can actually end up with killing the mice in the studies. So that each cost is actually thousands of dollars, and which actually, if you consider the amount of mice for a study, so that actually contributes to high cost. And the blood actually, when you take the blood, it requires usually a lot of steps before you get the detection, which makes the test time-consuming. And most of the tests only can detect single biomarker at a time. That actually also contributes to the turnaround time. So there is an apparent need for better diagnostic or detection tool in the industry. So to overcome these challenges, Biolom developed a platform technology that uses antibody-coded nanoparticles assembled on a semiconductor chip. So this is the test is done with the sandwich assay. The antibodies are coded on the nanoparticles. On the right side, you can see, and it binds to the protein of interest, and the tests are incubated by secondary antibody, following by which is attached to fluorescent tag, and the results can be obtained in optical microscopy, fluorescent microscopy. So this is not a unique thing in the Biolom, because it's a sandwich assay, is well known, it's traditional. The uniqueness Biolom has the unique expertise to manipulate and assemble these antibody-coded nanoparticles precisely on the locations of the chip, the semiconductor chip that we have. So that will actually, by assembling these nanoparticles, by the way, each nanoparticle can go down to 100 nanometers size. So by assembling these nanoparticles on a chip, you can actually, one can accommodate large number of antibodies in a very small area. That actually enables a very small detection area in the sensors, and very small sensor size actually. The sensors that Biolom makes is the total size of the sensor, the diameter is around 300 micron. The detection area can go to 100 microns. That will actually allow us to use this sensor in vivo testing in addition to in vitro. So what I mean by in vivo is this sensor can be attached on a catheter, or it can be attached at the edge of the needle that can go into the vein and wait the whole blood to circulate over the sensor, and the sensor can be taken out, and the result can be obtained in traditional fluorescent microscopy. So the other advantage is since we can accommodate these antibodies on specific locations, and we can align them and localize them in a very small region, the results are only obtained on those regions where we have the nanoparticles, nowhere else. So it's very high resolution fluorescent imaging that will actually increase the sensitivity of the detection. So I will show you the results. We can go up to 100 times more sensitive compared to what has been used right now in the settings, clinical settings and industry. So, and also not only that, we can also assemble more than one type of nanoparticles on the device surface, so that will allow simultaneous detection of multiple biomarkers. So the last but not least, the sensor uses a very small sample volume. Only finger prick the whole blood is enough for the detection. So that also has the very fast results. You can get the results in minutes instead of hours. So that the outcome of this technology is actually a small, very highly sensitive, and you can see the sensor already. The product has been developed. You can see on the left side, my colleague is holding the sensor on the catheter. It's holding in his hand. It's like 0.3 millimeter in diameter. For in vivo, this you can detect. This can go into the vein and detect the blood, the antigens from the blood, and it can go to multiplex detection. So this has been already, the technology has been published in many journals, and we have patents already granted, and we filed 2 more patents pending. So before I go to the results, I want to explain you how our sensor is different than the typical ELISA, which has been used right now in the clinical settings. So the detection technique, first of all, is high-resolution fluorescence. It's not a chemiluminescence as in ELISA. So that actually brings 2 advantages. One is you can use any optical microscope, which has a 500x magnification objective. And also you can have very high resolution imaging because you can focus on the regions and magnify those regions and obtain the signal from localized regions. So if you look at the ELISA, the antibodies are usually coded randomly because it's only a passive absorption on the ELISA plate, 96 well plate setup. However, if you look at the Biolom's technology, we use a carboxylic functionalized nanoparticles, which are actually increasing the likelihood of the antibodies to align because it's not only a passive absorption, it's also we take the advantage of electrostatic interaction. So that's one advantage. The second one is the ELISA is only single biomarker. It's a very large well. It's actually, you need to put 7 millimeter, it's a diameter is 7 millimeter. You need to put a lot of reagents, and it requires a large sample volume, and it actually contributes to high antibody cost. However, the Biolom sensor only uses a very small, because of very small detection area, it only uses 1% of the reagents that ELISA uses. That actually lowers the cost significantly. So the last point is, as I mentioned you, it's highly sensitive because if you're trying to measure very low concentrations over a very large area of the well, in the ELISA, it's a very large area. If you have very low concentrations, you only, the signal have very low from a localized region. However, if you look at it's localized, you only get a very strong signal only locations where we assemble antibody-coated nanoparticles. So that increases the sensitivity. So these are some of the results that we obtained. The in vitro results, where we drag we draw the blood and test it in CEA, which is a colorectal cancer biomarker. So we were able to get down to 15.6 picogram per ml in vitro, and we also injected CEA antibody and antigens in the mice, and we were able to get to around 31 picogram per ml. So this is, if you compare with the ELISA, this is more than 100 times improvement because the current cutoff levels of the CEA ELISA kits are 1,000 to 2,500 picogram per ml. This is the results from in vivo microchip, the small chip that I showed you before, and we implanted the chip actually in the catheter. We implanted to the mice, which is injected by CEA antigen spiked, and we were able to get down to around 25 picogram per ml. And we also demonstrated the multiplex detection on a single chip. So on the right side and the lower side, you can see this chip that has 4 areas, and each areas are incubated with different antibodies, 2C5, 2G4, and IgG, and we can have multiplex detection using this chip. So these are the experiments. This is the direct comparison with the ELISA, 96 well plate ELISA and Biolom's biosensor. Actually, in this study, what we did is we injected a tumor, the CEA that produced CEA antigens, and to the mice, and we actually monitor the blood level, the antigen level in the mice over a time. So we observed that the Biolom sensor was able to detect the CEA in tumor-bearing mice at day 7, whereas ELISA 96 well-plate ELISA that's commercially available was not able to detect. So this actually shows the potential for early detection of the biosensor. And as I told you, it's a platform technology. Doesn't only work in CEA. It doesn't only work for the cancer. We can have a different kind of the antibodies coded on nanoparticles, and we did this with nucleosome, which is a generic biomarker. We did this one with the PSA. For prostate cancer, we were down to 20 picogram per ml compared to 10 nanogram, which is the recurrent threshold. And we have tested not only cancer, but also cardiac myosin, 2G4, the antibodies, and we were able to get down to 250 picogram. So there are other technologies out there, the Biolom's competitors. I already showed you the differences between ELISA. I'm not going to go through again, but there are like MSD, mesoscale discovery, Luminex. They have very high sensitive detection. However, if you look at those technologies, it has various disadvantages over the Bioloms. For example, MSD uses electrochemiluminescence. It's a very complex. You need to electrically activate the lithium-labeled antibody. For that, you need to use a lot of streptococcus and DNA molecules to align the antibodies and to activate it. So this requires spatial reagents, large sample volume. Assay times usually it's hours, not minutes, as Biolom. And Luminex is the same. They use magnetic nanoparticles actually as countable, but these particles are not stabilized on the surface as Biolom. They are in the fluid, and this actually increases the agglomeration of the magnetic nanoparticles. That's a huge problem, and it actually affects the accurate detection of the biomarkers. So if you look at Biolom, we have the multiplex and very high sensitive detection. And by the way, none of those competitors can do in vivo testing. So Biolom has a very small in vivo, and you can have very small sample volume and get the results from the finger prick. So the competitive advantage chart shows where Biolom stands on the right and upper side. It has a high analytical sensitivity and lower cost. In addition to this, Biolom has the license with exclusive rights on the first micron scale multiplex sensor in the market. So we think that this is this will have a very huge competitive advantage over our competitors. So so far, I showed you the technology, competition, and the market. We had this technology and we wanted to explore what are the potential customers and where can we use this device. So for this one, we went through a program with NSF, I-Corps. We were awarded $50K for customer development. Just during this program, we interviewed over 70 potential customers. So some of them were face-to-face, some of them were on the phone, and we went through this program in 2 to 3 months, and we get very good feedback about the potential users. So in the next couple of slides, I'm gonna explain you what were these the potential customers. So one insight was the use of our sensor in drug development in clinical trials. This could be mice studies, or this could be the human blood testing in clinical trials. In mice studies, as I also mentioned to you before, can use this device in vitro testing, which requires only 10 micron blood from the mice. Instead of right now in current settings, you need to throw almost 5 to 10 ml from the mice that actually end up with killing the mice. And as increases the cost, especially if you are doing a very advanced studies with genetically identical mice, for example, that each mice can cost to tens of thousands of dollars. So considering how many mice you need to study, it's a lot of cost. So using our in vitro settings, you can draw only 10 micron blood without killing the mice, and this will actually bring you a major cost and time-saving advantage. And also, our sensor can be used in in vivo sensing. The sensor can be placed at the tip of a needle, and again, you don't need to kill the animal, and can get access to whole blood circulation. Again, major cost and time-saving advantages. So, one step forward from the animal studies are the human clinical trials. So this is in vitro testing right now. Instead of having a 5 to 10 ml from the patient, you can increase the patient convenience, and you can have sample from a finger prick, and that will actually can increase the turnaround time of the testing during the trials. So this was the one aspect. The other aspect is the diagnostic. We get very good insight from the diagnostic companies like Roche. We talked to Roche, and they suggested to develop us a compact and easy point-of-care reader that can actually enable instant results in a fully automated device. We already have some companies talking right now to develop this reader. And also we talked to the clinicians in Boston Medical Hospital, and they said that this test can be since it's very highly sensitive, it can be a very good complementary test for the CyberKnife test, the radiotherapy test that they've been already carrying out in their hospitals. So what they do is actually they kill the tumor using the radio waves, and they send the patient to the home, and they monitor after 3 to 4 months. So the reason they are waiting for 3 to 4 months is they are waiting the tumor, its potential to grow so that it can create enough antigens in the blood, and that can be detected using a current ELISA. However, our tests are so sensitive, you don't need to wait 3 to 3 months. Even the tumor is very small and it generates small amount of antigens, you will be able to detect using our. So this is a nice example of how the diagnostic and the therapeutics actually can go together using our sensor. So, and also contacts, we talked to Dr. Lek Bakar in Beth Israel Hospital. He actually pointed out the need for early detection of liver tumor markers in high-risk groups. So these groups are and hepatitis C and hepatitis B patients. It's already more than 5 million patients that has the potential of having a liver cancer. So he said that our tool is so sensitive, and we can use, specific, you can actually use this one. He can actually use this sensor to screen these patients to see if they generate any indication of liver cancer. So the Biolom product concept, we are envisioning the product as I already showed you, it's a semiconductor chip with the sensor area. So we are envisioning it like a cartridge that can actually, the sample from a finger prick can be put onto the device, and that can go into the reader device. So that the reader can have the single biomarker test, multiple biomarker test using our sensor. So we have the Biolom team here, and if you look at the team, it's very interdisciplinary. It has people from different fields. For example, Jay Devaponi, he's a PhD in pharmaceutical science. He's specialized in incubation of nanoparticles with antibodies. And me, myself, as a mechanical engineer, and I actually, my specialty is precisely assemble these nanoparticles on the surfaces. And Asanta Malima is a PhD in electrical engineering. He's a device fabrication, expert in device fabrication and microfabrication and sensors. And we have also George Ranuk. He's an MBA in mechanical engineering. He's bringing a business experience to the company. He has raised 4 startups, over 30 years experience in operation in test manufacturing. And we have also a very strong advisory board team. Dr. Ahmet Bosnena on the line. He's the director of NSF Center. His expertise in semiconductors and nanotechnology. And Martin Streeter is a co-founder of STAT in life science, and he has also he's actually a former investor. He's bringing a lot of insight to the team. And Dr. Vladimir Torchelin, very well known in pharma, in drug delivery, antibody incubation. He's currently at Northeastern University. And we also collaborate with doctors in Mayo Clinic, Boston Medical Center, and Beth Israel Hospital. So the progress to date is actually, I want to summarize in this slide to progress and want to mention about the future milestones. And the technology has been developed. We proved the concept in animal blood, and almost more than $2 million, as Professor Bosnena explained, has been spent using Keck Foundation and NSF Foundation on this research. And we incorporated the company last year. We made several progress, received a lot of awards, and raised over $80,000 from private sources. And we have secured the license agreement from the Northeastern University. So the future milestones, we are really looking to partner with pharma diagnostic companies to extend our animal studies. This could be monitoring the effectiveness of the therapeutic agents. And also in the future, we really want to test the sensor in human blood in clinical trials. And this could be, as I told you, this could be a more accurate clinical trial test to evaluate the remission of a cancer or any other disease. And the last milestone, actually, we are working on it right now. We are developing talking to companies to develop a fluorescent-based reader that will actually very advantageous in the future if we want to take this one to the hospitals, and because it can, it can be a benchtop device in the doctor's office, could be a benchtop device in the clinics, in reference labs, for example, that could, enable a very fast turnaround time for the sensor. So with that, I would like to conclude my talk. We are happy to answer if you have some questions. Thank you.
Great. Thanks very much, Cihan. That was a terrific overview of your technology and provided lots of details. If you could just flip the, make me the presenter. Thank you so much. So normally our scientific director, Dr. Ernie Bush, would lead this part of the discussion, but he was unfortunately double booked for today, so I'm just going to open it up and take us through a couple of questions and some thoughts here. So this is really an opportunity for the industry members on the line. We have Terry, we have JMR, we have PN, and we have MR. To ask any questions to either Cihan or to Dr. Bosnani to explain, the technology, talk about applications, and maybe what I'll do is I'll just flip back to, well, there's Dr. Bosnani and there's Cihan. Just explain anything or ask any questions. All you have to do is unmute yourself, which is to the right of your name, and then I'll see that you're unmuted, and then I'll say, yes, what would you like to ask, or you can just jump in. And for you, PN, if you want to put any questions into the chat box, then I'll just read out your questions and get them to answer them. So don't be shy, because I can't come up with the questions that you might have, because you're looking at how this might help move science forward and clinical trials forward faster and better in the pharmaceutical industry. I do have some prompting questions for you guys if you don't have any questions for Cihan or Dr. Bosnani. Ahmed, Dr. Bosnani, do you have anything that you'd like to say as summary comments right now?
Yes, thank you. We intended for this in the beginning to be a diagnostic tool for patients that would be very accurate and uses whole blood and uses a tiny amount. And we developed an in vivo biosensor because of its size. You know, the whole sensor size is 100 micron, for example, and it was designed to be glued to a catheter. We developed a whole assembly station, fully automated to assemble, to take that small sensor and actually glue it, do the assembly of the particles and glue it, completely guided by software. And this was supposed to be integrated with either a needle drawing blood or an IV, for example. So it'd be touched with the blood. But then after we talked to a lot of people in companies, they indicated that since we've done this, we use this with mice, and since the catheter and the sensor fit into the vein of a mouse, that means they don't really have to kill the mouse. And even if they use it in vitro, it's a very small volume of blood that's needed. That the study can, whatever drug testing studies that are done with very expensive mice, it means that you really don't have to kill the mice for the whole study. You can use follow the same group of mice, which will be a huge advantage cost-wise, but also more accurate because you're not actually having data points from different mice as the study goes on. You're using the same mice all the way through. And as Jihan showed, that the mouse that we have used for where the cancer cells were injected under the skin of the mouse, we saw a small indication signal after 3 days, but a very definite signal after 7 days, where the ELISA did not really detect anything until the tumor was very visible and very large, for example. So it turned out that it can be used for diagnostics, like he mentioned, especially for people in remission with cancer, because it's very sensitive, and you don't have to wait for 3 months or 6 months to come back and check for to do a CAT scan or MRI to check for cancer. But you can actually, or do a blood test, you can actually do it within the same day after surgery. So it seems like it's a single platform that actually can be used for a variety of things. And as you mentioned, we've used cardiac biomarkers as well as cancer biomarkers, but actually it's an antibody-based technique, just like the ELISA is. And so it really can be used for a variety of biomarkers that have antibodies. So it can be used for a variety of things. So we're hopeful that this can have wide use, especially initially for drug development, and then later on with approval, FDA approval, and so forth for diagnostics. Any questions?
Great, thanks so much.
Thank you.
So I'm going to ask the audience a couple of questions directly. And so I'm just wondering if anyone who's online today has any experience with this sort of system, or comparable systems, or this technology itself. Have you used this sort of model, or know of its use, or evaluated it for in-house use? So does anyone have any thing that you would consider comparable or similar to this that you think would be a comparator as we go forward in evaluating this sort of technology? And maybe I could ask PN to put your answer in the chat box, and maybe I could pick on Terry and JMR. So while I'm waiting for them to think about that, I do have a question. Please clarify, how is fluorescent intensity accurately measured with this system? So that's for Chihan or Ahmed.
Well, this has to be calibrated, just like the ELISA technique. So there is a calibration that goes in, and typically with a sensor, we have a reference fluorescent area that can be used as a baseline, for example, to tune your fluorescent microscope to a certain baseline. It's very similar to the ELISA technique, except it's much smaller, and you have to use more magnification because it is smaller. It is a smaller area. than the well area that's used for the ELISA.
Okay, great. Thank you, Ahmed. So is there any other additional information that the participants today would like to have in order to evaluate this further, or any other additional next steps that you think should be done besides the ones that were listed in the previous slide? We have one comment here from, other than ELISA, JMR has said we have not used a similar technology. So in order to move from ELISA to this sort of technology, what do you think it would take?
So I'm not sure I understand the question. Is that, I can answer your question, Don.
Okay.
So basically, I mean, one of the reasons, I mean, we have other technologies, but one of the reasons we chose the ELISA because everybody's familiar with that. And so there was no question about how to operate. But we wanted to make the ELISA 100 times more sensitive, and want the ELISA to use whole blood, and want the ELISA to be the volume of blood that's used to be really tiny, much less than what used for a glucose, Bert test. And also, we wanted to make the ELISA in vivo. So we accomplished these 4 different things. And if somebody wants just to use it in vitro, then it simply, can just use a blood droplet, on the chip, for example, and that would be sufficient, for example. So it has a lot of different uses, and it did accomplish 4 things that the ELISA doesn't have. And we wanted this to be, right now we do it on a fluorescent microscope, but this can be easily automated with a reader. It could be desktop, and it could be eventually with the an attachment to an iPhone, for example, that have a microfluidic chip that will do the reading the signal and reporting the signal.
Great. Okay. Go ahead. Someone wanted to say, Jihan?
Yeah. I want to add something to here. As Professor Busnera said, so this is right now we use an optical microscope, which is a typical optical microscope, which is available in any labs, any research labs or company labs, as only require 500x objective. So you can actually test this one, have a fluorescent So, but as Professor said, it's gonna if you want to have this one in the reference labs in the future or hospital setup, it needs to be automated, and it can be easily done because the fluorescent technique is not very different than what has been using right now.
And I know you are comfortable with the idea of the hospital setup, but of course, what I think you're referring to, Chian, is the clinical trials done in a hospital or clinical setting. Yeah.
Yes.
Okay. Another comment here coming in from the group: the technology is very interesting. We're using Luminex and /MSD extensively in our group. It will be good to see your claims compared to, say, for example, multiplex detection of cytokines in the blood. So any comments about that? And then I have another comment that's coming. So to Ahmed and Jihan, any comments for you about what?
We have not. So can you read the question again, please, Tom?
We are using Luminex /MSD extensively in our group. It will be good to see your claims compared to, say, for example, multiplex detection of cytokines in the blood.
Oh, the cytokines. Okay, so so first of all, Luminex is, it's a magnetic bead incubated nanoparticles. It is it's mostly the accuracy is like sensitivity is not very it's questionable because most of the time it's just nanoparticles are because nanoparticles are not stuck on the surface as we do, and they actually travels in the liquid that actually increases the agglomeration of the particles, and most of the time it can be costlier the equipments that detects this tests can be costlier. So we we can just however in Biolom it's very different, and we have a high sensitive and the cost, and also, by the way, you can just the comparison with the Luminex, the biggest advantage is the droplet of blood, from the finger prick. We use whole blood from the finger prick to detect these biomarkers. Cycloteins, we have not tested the cycloteins, but as I showed, my previous slides, we have tested the in vivo sensor with different biomarkers, 3 or 4 biomarkers on the same chip, and these are different biomarkers on the same chip. So so it's a platform, so we can just we haven't tested cycloteins, but we can just I believe it's very possible that we can just incubate with the antibodies towards the cycloteins and have the multiplex detection on the same chip too.
Sorry, great. I'm just moving back to the other slide. So another question is, a very interesting technology. Are there plans to use this technology to look at non-protein biomarkers also? It seems that nucleic acids could be used for detection of microRNAs, similarly to in situ hybridization.
Can you read the molecule? Can you repeat the molecules? Which molecules? It's
sure. So let me just stick it in the chat box as well. So are there plans to use this technology to look at non-protein biomarkers also? It seems that nucleic acids could be used for detection of microRNAs, similarly to in situ hybridization.
Yes, nucleic acids can be used. So I mean, in order to use this technology, whatever we can bind on the nanoparticles, right? It's a passive absorption with the electrostatic interaction. That's how we bind the antibodies or other molecules to the surface. So as long as we can bind them onto the surface, we can assemble these nanoparticles on the nanoparticle surface. We can assemble these nanoparticles on the device surface and detect, for example, what they are suggesting, RNAs or any other. That's why we call them as a platform technology. It's not really limited to 1 or 2 molecules. It can actually work with many of molecules as long as we can bind onto the nanoparticle surface. And most of the molecules, actually, there is a specific, you can use, reagents and some of, intermediate binders to bind these molecules. So I believe this is possible. Yes.
Okay, great. And Brett Johnson, who is part of our team and used to work at Novartis and some other companies, he just wants to jump in and add a point there.
Yeah, I think circulating nucleotides and circulating DNA would be an easy step for this from the whole blood aspect. I think when we're talking about cell lysates, I think that's another thing that can be tested and validated fairly easily, especially when we're talking about in situ type of hybridization.
Great. Thanks, Brett.
Mm-hmm. That's a very good suggestion.
Another question that's come in here, and I think everyone can see it, but I'll place it in there again. Do you see this technology being used primarily in preclinical testing with ELISA technology, that is Luminex, being used for validating and application to clinical studies? Or could this technology be validated for clinical use? And my guess is you're going to say yes.
Mm-hmm.
And maybe you could just
both.
explain a little more.
Yeah, you see it. So I think it's both. So this is as I mentioned in my slides, this could be preclinical testing in animals. We can use the in vivo sensor again as the advantage of not killing the mice and cost advantages and turnaround time, but also it can be used in in clinical patients trial. Can be only finger prick the blood from the finger prick could work by using our sensor in vitro testing.
Okay.
So that's actually our next step. We validated the technology in mice. Actually, we didn't have extensive studies in mice in the research environment. But first, that's why we want to extend our preclinical testing in animals. Then we want to move forward with the clinical use and in clinical trials in patients.
Yeah, and I might add is that we have done the animal studies, but we have not done human blood yet. And this is much easier than Luminex. I know Luminex is being used, but Luminex you have to do the assay, and then the particles have to be added, pipetted, out for readout, and then it's really very laborious compared to a chip that already has the particles on. That all you have to do is just apply the whole blood on top of that, and then wash the blood, and then put the solution with the secondary antibody with the fluorescent tag, and that's it. That's all you have to do. And you're ready to read the signal. There's no agglomeration involved. There's no matrix interference. It's very specific. Very cheap compared to Luminex, and there's no moving particles at all, or the particles are locked in, for example. So it has a lot of advantages, but it still needs to be approved for, and verified and validated for clinical trials with human blood.
Great, thanks, Ahmed and Chihan. So a couple things. One is we've put the contact information for both Chihan and Ahmed into the chat box if you're interested in talking to them directly afterwards. Also, just to remind everyone that we really need you to fill out the survey. And I'm going to turn to this one question here as the last question. If you guys could let me know from the industry, typically we would kick the Northeastern guys off and see if there's anything that you want to ask privately with them not on the line. If you can just let me know if you think we need to do this, or if you think all of your questions have been covered by you sending me private questions and then me reading them out. So I'll read out this last question, and then Chihan and Ahmed, you can answer it. We're using MSD currently, but have seen some unexpected variability at times. It would be very interesting to compare the variability of this technology versus MSD. Have you made any internal comparisons of your assay system versus MSD for any particular endpoint to see if you have improvements in consistency within the range of both assays?
No, we have not done a internal comparison with MSD, but as I mentioned, the MSD is a very different technology. It's electrochemiluminescence, and it introduced a lot of matrix interface, non-specific bindings, mostly that could actually turn out to variability in this test. And no, we have not done it, but this can be done, but also, as far as I know, the MSD doesn't use a whole blood, so that's another thing. So in order to do this, we have to use what and we compare. It's better to compare with one-to-one. If you have one-to-one comparison, can be compared with the serum. So no, the short answer is we have not done it, but it's a very, the MSD, the way that they did detect, it's possible that they will have variability because of the matrix interface and other problems that I mentioned. Yeah, but it's doable. Yeah.
Okay, great. So I've got one message that says that we don't need to go offline. Just a reminder, we would like everyone to submit the surveys. We've put this survey link up in the chat box again. If you click on it from the chat box, it'll take you right there, and it should take you about 5 minutes to answer the survey. But if you can't do it right now, we'll send you out the link when we send this recording, and we would like it to have it back by Monday. But if there's other people in your company that you want to share it with, feel free to share them the webinar so that they can listen to it as well. Any other closing remarks from either Chihan or Ahmed?
No, thank you very much for this opportunity, and we really would like to, it was great feedback, and we are open to, the discussions. Please contact us if you have further questions or if you have any interest, partnering, collaboration, any topic. As I mentioned to you, right now, the sensor is ready. We have developed the technology. We manufacture these sensors at right in Boston, so we can just, looking forward to partner and collaboration with the pharma industry.
Great.
Yeah,
Go ahead.
I think that that's exactly what we're looking for, because we developed this technology. I think it's right now, we think that probably the first step is to use it for development with animals and so forth, because it's very accurate and doesn't require killing the animal, for example. It's very fast. But also that can be used for clinical trials, and then eventually could be used for with actual patients for point of care. So we welcome any input. We certainly welcome any inquiries, or we're looking forward to any desire to partner. For example, we're looking to partner to develop this technology further. And we think it does have a lot of promise compared to what's available right now. And thank you.
Great. Well, thanks very much to everyone for your participation today, for the presentation today from Northeastern and Biolom, and for the interesting questions and thoughts that everyone provided. So again, this has been recorded. We'll send you the link. We would love you to fill out the survey and get back to us so we can continue to evaluate these technologies and share them with the rest of the industry. So again, thanks everyone. Have a great day. And if you have any questions or suggestions for improvement, we're always open for those as well too. Thanks a lot. Take care. Bye now.
Thank you.
Thanks.